So let's look at the structure of nucleic acids. We're going to start off with the primary structure. Well, each nucleic acid has its own unique sequence of bases that carries the genetic information from one cell into the next. So nucleic acids are polymers of many nucleotides in which the 3' hydroxyl group of the sugar in one nucleotide binds to the phosphate group on the 5' carbon atom in the sugar of the next nucleotide. And they each have a unique sequence of bases, and this is called their primary structure. They have one terminal end with an unreacted or free sugar with a 5' phosphate, and they have a sugar at the other end with a free 3' hydroxyl group. So when I talk about the primary structure, I teach them as a pearl necklace. Okay, so it's like a pearl necklace with all the individual pearls in a strand next to each other. Okay, so this is going to be your primary structure. The link between the sugars and the adjacent nucleotides is referred to as a phosphodiester bond. Sometimes this is called a peptide bond as well. So kind of remember your P's for that primary structure. It's like a pearl necklace. It's the primary structure with those phosphodiester bonds. Okay, so again, you can see that you have one nucleotide here. You're linking it together with a phosphodiester bond. And then here is going to be your other nucleotide, right? And you're going to have the free 5' phosphate group on the one, and then the free 3' hydroxyl group on the other. So looking at the study check, let's draw the condensed structural formula for the RNA nucleotide that's formed by 2 cytidine 5' monophosphates. That is a mouthful. All right, so pause me and come back. Well, the dinucleotide is drawn by connecting the 3' hydroxyl group on the first cytidine 5' monophosphate with the 5' phosphate group on the second. And so what you would end up doing is you would combining it here, and so then you have 1, 2, making it a dinucleotide that has 2 cytidine 5 monophosphates phosphates combined together. All right, so moving on with the nucleic acid sequence, the sequence is read from the sugar with the free 5' phosphate to the sugar with the free 3' hydroxyl group. And the sequence is written using the letters of the bases A, C, G, and U. And remember from our last video, since we have a U, that means that we are going to be in RNA, not in DNA, because DNA has T's, A, T, C, G, where RNA has the U's. Okay, so since we see a U, we know that we're in RNA. All right, so fill in the blank. In a nucleic acid, the nucleotides are joined by what kind of bond that joins the 3' hydroxyl group of the sugar to the phosphate group on the 5' of the next nucleotide. Pause me and come back. So remember that is called a phosphodiester bond. All right, great job. All right, in the nucleic acid CGAU, which base is at the free 5' phosphate end? Pause me and come back. All right, the correct answer is going to be A, which is cytosine, because cytosine is going to be at the free 5' end.